Well, good afternoon, everyone. I would like to welcome you to this special plenary presentation on microbial life in the cosmos. Is this microphone on? Yes, I see if I can do a better job of speaking into it more properly. It is indeed my privilege and honor to introduce the first speaker of the afternoon. Everyone is well aware that about a year ago, Dr. David McKay and his co-workers made an announcement of the discovery of possible relic biogenic activity in an ancient meteorite from Mars and possible microfossils associated with that meteorite. Dr. McKay is manager of the electron microscope laboratories at the NASA Johnson Space Center. He founded the electron microscope and microprobe labs along with the Lunar and Mars Resource Utilization Program. Dr. McKay received his BA degree from Rice University, MA from the University of California, Berkeley, and PhD from Rice University. David McKay has received the Principal Investigators Award, a Geological Society of America Award for astronaut training, as well as numerous NASA awards, including the Achievement Award. The presentation he will give this afternoon is entitled Search for Past Life on Mars, Possible Relic Biogenic Activity in a Martian Meteorite. David McKay.
Thank you, Richard. What I'd like to do today is briefly summarize the data that prompted our original interpretation and then move on to some new data that we have, most of it not yet published, but some of it published, and then finish up with time for questions and answers. So if you can, let's go directly to the first 35 millimeter slide, I guess, and the lights, please. Well, I want to point out that our team is in fine print here, but we do have a very nice team, including people at Johnson Space Center and also at Stanford University. And our team originally was at one of those two places. It's now scattered a bit. One member has gone to McGill, and one member has gone to Savannah River. Well, everyone has to show a slide of Mars' surface. This is an old Viking slide, but I like it because it clearly shows that there are rocks there, there are different colors on the rocks, and there are different colors in the soils, that it's kind of a complex place, even in these photos of the surface. But the problem is, we just can't tell much about what's inside those rocks and those soils. And furthermore, the Viking results, with the exception of possibly of the labeled release experiment, which Gil Levin talked about this morning, were mostly negative in terms of the possibility of life on Mars. And so that subject lay more or less dormant. There was a lot of controversy about it for a while, and then it got pushed aside on the shelf for a number of years until we started to realize that some of the meteorites in our possession were actually from Mars. And at first the modelists said that's absolutely impossible. You can't get a meteorite from Mars here without melting it and vaporizing it. If you got any here, it would be glass. In fact, as this artist's conception shows, there are ways to get meteorites up out of the gravity field of Mars without appreciably shocking them. And now the modelists have gone back to the drawing board and worked that out and allow it. Why did that paradigm change? Because we started finding in Antarctica meteorites, first from the moon and then from Mars. And I'll tell you in a minute why we think they're from Mars. Allen Hills, that I'm going to talk about later, is right in this area. It's an area of blue ice, which I will I will talk about. And what happens in Antarctica, it's really nifty. Meteorites fall on the surface of the ice. The ice slowly is flowing downhill from the South Pole, radially out toward the sea. Occasionally, in some areas, the ice encounters an obstacle such as an underground buried hill or mountain area, and then the ice tends to come up. And as it comes up, it's got nowhere to go, and so rather than piling over the mountain, it's exposed to these very high velocity, very dry winds, the katabatic winds. And it is, in fact, eroded away at the rate of a few centimeters a year. As it erodes away, it exposes meteorites. Now, the area there, this is a summer day near the collection, one of the collection areas, and you can see that there's quite a bit of wind and still quite cold, but it is this wind and the cold, dry atmosphere that really helps us because it essentially exposes meteorites. And in fact, more than 90% of the rocks that you see out on the ice are meteorites. You don't see them everywhere you look, but if you look around for a while, you'll see a rock, sometimes even from a helicopter. And when you go to that rock, it turns out to be a meteorite. This one is on a pedestal, it's sticking up. The rock itself has prevented the erosion of the ice directly beneath it. These rocks are collected every year by a joint NASA NSF expedition. The Japanese have a similar expedition, usually to a different place, hopefully, so they're not competing. These rocks are brought back, and it's incredible. They typically bring back from 300 or 400 to close to 1,000 meteorites each year. It has essentially increased the supply of world meteorites by about a factor of 10 since the program has been running. This is the first lunar meteorite. This rock comes from Antarctica, it comes from Allen Hills, and it was recognized after careful work that this was from the moon. How did we know that? Because we had studied lunar rocks for a number of years, and when we saw this, it was just a dead ringer for a lunar rock. Mineralogy, chemistry, texture, things like oxygen isotopes, things like iron-manganese ratio, all dead giveaways that this was a lunar rock. No doubt about it. And yet this rock, although it had a glassy matrix, was not shocked beyond recognition. In fact, much of it was relatively unshocked. So this was the first clue that meteorites could come back, come to us from another planetary body without being destroyed. And then the meteorites all the Antarctic meteorites are brought and processed in clean conditions in Houston. And in fact, this is a meteorite from Elephant Moraine, which we call the Rosetta Stone of the Mars meteorites, because it is this meteorite that enabled us to make the very close connection between this class of meteorites, which has previously been known as SNCs, SNCs. We made the connection between that class and Mars based entirely on this meteorite initially. Other data now support it. But see these little round areas? These are little pockets of glass within this rocky matrix. When those pockets of glass were analyzed, it turned out that they contained rare gases here along this axis. All these rare gases were analyzed, and we already had an analysis of the Mars atmosphere from Viking. And lo and behold, the match was nearly perfect, over 8 orders of magnitude. The match between the abundance of gas in those little glassy pockets and the abundance of gas on Mars, as measured by Viking, was a totally close 99.9% correlation match. And in fact, that particular abundance pattern fits nothing else in the solar system that we know of. So we can immediately eliminate Venus, we can eliminate We can eliminate Titan, things like that, have totally different atmospheres. So that was the first real proof that this meteorite came from Mars. Subsequently, other meteorites have been analyzed for gases, and rare gases matching Mars' atmosphere has now been found in at least 3 others. Now, once it was realized that one of these guys was from Mars, what about the other ones? Well, it turns out that this family of meteorites called SNCs, SNCs, all fit in a fairly tight envelope here on a 3-oxygen plot. This is a way that plots oxygen isotopes, and in fact, all the terrestrial rocks fall on this line, and there's a line up here clearly displaced from the terrestrial line. And the conclusion from this diagram plus the previous diagram, plus a number of other detailed bits of data, the conclusion is that, hey, if one of these is from Mars, they all got to be from Mars. So if that Rosetta Stone, that 79001, is from Mars, then all the others, including 84001, which I'll talk about now, also have to be from Mars. That conclusion is accepted now by about 95% of the meteorite community, that these meteorites are from Mars. There are a few holdouts. The fact that 95% accept it doesn't make it true. It's just the way consensus tends to develop. Well, so where are we now? These are the Mars meteorites. So 12 of them total. Half of them are from Antarctica. The ones that are not from Antarctica have mostly been observed to fall from the sky and were picked up immediately. This table goes back to 1803. I can't quite read it, but it's that was the first one, and then 1815, I guess. That was the first one, and then throughout the past nearly 200 years, others have been observed to fall. They all fit on that line shown in the previous graph. Allen Hills is the one I'm going to talk about now. It's almost 2 kilometers. It's not the biggest by any means. This is it. It's not particularly photogenic. It's got a fusion crust that covers a good part of it, somewhat eroded away in places by the wind in Antarctica, presumably. The rock itself is a pyroxenite. It's mainly pyroxene, which is a calcium, magnesium, iron silicate. And the rock was passed out to a number of different laboratories. And here's what we know about it. It's been carefully dated by 3 different techniques. It is about the oldest rock that we have. It's older by far than any Earth rock, and there are a few lunar rocks that approach this age. But it's probably the oldest well-documented rock in existence in our laboratories. The carbonate that I'm going to talk about is younger. It's very difficult to date. The current dates by different laboratories, 3.1 to 3.6 billion years old. We know that it was removed from Mars by that big impact shown in the cartoon earlier about 16 million years ago. How do we know that? That's based on radiogenic isotopes, which are activated only when it was in space and exposed to cosmic rays and solar flares. And there are three different techniques, all agree within a couple million years of about 16 million years. That's the time it spent lost in space. It didn't come directly to Earth. It orbited around the sun for most of that time, and it could have gone further out in the solar system and then worked its way back in. The orbits of these ejected meteorites are very chaotic, and they're influenced by gravity of Jupiter and all kinds of things. We do know that it landed in Antarctica based on carbon-14 age dating of carbon in the meteorite about 13,000 years ago. It was found in '84 and somewhat misidentified, but David Middlefelt finally realized in 1994, only 3 years ago, that it was a Mars meteorite. And then it was passed out for further study, and we got our sample early in '95. Now, there are a lot of interesting things about this meteorite, but one of the things that attracts everybody's attention are these little features. These are carbonate globules. They're carbonates that's about 0.2 of a millimeter across, and they have an orangish brown core, and then they have a rim consisting of black, white, black. We sometimes call that the Oreo cookie rim because of that sequence of black, white, black. The black stuff is really iron carbonate. The white stuff is really mainly magnesium carbonate, and then we go back to an iron carbonate rim at the very end of it. This is a backscatter electron photomicrograph made of it with some false color added, and again the cores are the orange. The black, white, black rim is now reversed because iron has greater backscatter electron emission efficiency, and then the basic green matrix is really the pyroxene, clinopyroxene. All right, now I'm going to concentrate for a while on these carbonates. We realized that they were unusual. We said, how did these guys form? What's going on here? This is just a cartoon to show the core, the rim, and most of them are rather flat. And in fact, they fit into pre-existing cracks in the meteorite. The surfaces tend not to be so flat, but are often undulating, and that corresponds to the crack shape from which they came. So it seems clear, most have concluded, that these things formed in cracks in the igneous rock. So one of the first things we did was map these with an electron microprobe, which can be set up to look at different compositions. This is only iron here. Everything you see, every bit of variation you see is a variation of iron. If this had no iron, it would be completely black, as is this little bit of mescalinite here. The carbonate globules, again, have a lot of iron in the rim, but if you look closely, you can see there's a very complex zoning where the iron goes up and down a bit. This is the very same view, but now we're set up to look at manganese. Manganese is concentrated in the cores. Almost no manganese out here in the outer parts of them or here on the pyroxene. And then this is sulfur. Sulfur is very interesting. Sulfur tends to go along with the iron in the rim. And there's a little bit of sulfur in the center, and there's a bit of sulfur in these smaller patches out here, but not much sulfur in the pyroxene, almost none. So sulfur is clearly associated with the carbonates, but tends to go along with the iron. Now, what caused these things? We scratched our head, and we investigated a whole lot of ways to form this. We went to the literature, we looked up all kinds of carbonates, we looked up phase diagrams. We finally decided that these could not be formed at high temperatures because they just don't fit the equilibrium phase diagram. And that's been a point of controversy, but we evaluated that origin early. The thing that we found that matched best the properties were carbonates which were formed in solution at relatively low temperatures with the help of bacteria. Now these are some that were formed in the laboratory by these people at the University of Houston, and they formed them using some water from the Gulf of Mexico, which had bacteria in it. They sterilized the water and could not get these to form at all. And when they etched these, again, these are the scale of these is about the same as the smaller size of the Martian carbonates. When they etched them, they got something like this. These are also chemically zoned, and the part that sticks out the most are the most iron-rich parts. Iron carbonate is much harder to etch away with HCl than calcium carbonate. And so these things are clearly show some kind of complex zoning, and the zoning is on about the same scale as the zoning we see in the Martian meteorites. Now, does this prove that the Martian meteorites were made by bacteria by a similar process? Not at all. We never said that, but that interpretation is consistent with this data. In other words, we have a model here which fits our data, and the model comes from a true occurrence in the laboratory of bacteria forming these carbonates. Well, then we decided to look at these more. This is our co-worker, Kathy Thomas, looking with the transmission electron microscope. And what we do is we try to pick out some of these carbonate globules and embed them in epoxy and then slice them up with a diamond knife, a microtome, and we get sections that are 800 to 1,000 angstroms or 100 nanometers thick. And we put those in the microscope and shine electrons through them, and we can get three things. We get an image of the grains that we're shining through. We get electron diffraction information from the grains, which will help us identify the crystallography. And we also, the last thing we get is a chemistry. We have an X-ray detector with all modern microscopes have X-ray detectors, and you can detect emitted X-rays to identify the chemical elements in the sample and even quantify them. So that's what we did, and we were intrigued with these rims, so we started looking at these rims here. You have to pick an area and then very carefully track it down so you know what you're looking at once you get up to very high magnification. Well, in the iron-rich rim, we started to see little grains like this and this, little dark grains, and they were all over the place. And when we looked at them more closely, each one of them turned out to be a magnetite crystal, Fe3O4 magnetite, and they tended to be on the order of 50 nanometers, sometimes 100 nanometers in overall dimension. Some were somewhat squarish, others were roundish, a few were teardrop shaped. What we did not find in the rim were whiskers, long, skinny magnetites. We went back to the literature and we looked around and we found that there's a lot of magnetite described in the literature. This is from one of the literature references compared to ours here. And that magnetite in the literature that we could find that best fit was magnetite known to be produced by bacteria. Bacteria produce magnetite in two ways. First, they make some inside their bodies here, and that magnetite is apparently used by the bacteria as a sensor and as a method of locomotion to get to the correct environment where the most food is. And so these bacteria tend to follow magnetic lines of force on the earth to get to more favorable food locations. And notice they're about the same size as what we saw. Now there's another class of bacteria which makes a lot of magnetite outside of their cell bodies, and they just make piles of it, jumbles of it, and it is, that magnetite is made not for directional purposes, but for the purposes of energetics. They're either oxidizing or they're reducing iron, and that magnetite is more or less a byproduct of that process, and they get energy from that reaction. So in comparing our magnetite here, we saw, compared to earth bacteria, very similar crystal shapes. The size range was about the same. The chemistry was the same. We saw no sign of other elements such as titanium, and that's true of earth bacteria, magnetite produced by them. And of course, the earth bacteria have to live in relatively low temperature fluid environment. We had deduced on other reasons, for other reasons, like the chemistry and the zones that I showed you in the chemistry, that hey, these guys could not have been at high temperature, or those chemical zones would have been erased if they'd been hot for any length of time. We also deduced that the carbonates were out of equilibrium, again, something that is not likely at high temperature. So, in summary then, the magnetite is a very good fit for Earth-produced magnetite. Now, does that prove that the Mars magnetite is produced by bacteria? Not at all, but it's permissive evidence. It's a reasonable explanation. It allows us to make that inference without any major contradiction in terms of what we have described and the possible origin of that magnetite. Well, we did some other kinds of analysis. Our friends at Stanford did laser desorption analysis, and what we did here was take some of those same surfaces that had the carbonate pancakes on them and essentially scan a laser back and forth over those surfaces. The pulse laser scanned over the surface a few microns at a time, and as it scanned, it essentially rather gently liberated molecules of whatever was there. And then there was another laser that essentially hit it. This laser was tuned to band frequencies of PAHs, polycyclic aromatic hydrocarbons, and it would tend to ionize the PAHs and then they would go off into a time of flight mass spectrometer. This is a very sensitive technique which Dick Zare and Simon Clement have perfected at Stanford for analyzing, first liberating gently in this plume, and then followed by a couple microseconds with another laser that ionizes them and zaps them into the mass spec. So using this technique, we studied a large number of surfaces, and we consistently found patterns that are something like this, with 4 major peaks of these polycyclic aromatic hydrocarbons, and then a higher mass envelope of some other peaks. And this is quite different from many kinds of terrestrial PAHs spectra. The closest thing that we see to it are some of the carbonaceous chondrite meteorites. But even the carbonaceous chondrite meteorites, and this is a whole bunch of them here, and this is our Allen Hills up here. Even those meteorites are separated when we use a mass-weighted average. They are separated from all of the carbonaceous chondrites that we analyzed. And then the other bit of data that we got was the distribution of those PAHs, and in particular, as we went in past the fusion crust, which was formed as when the Mars meteorite came through the Earth's atmosphere, as we went past that, we start out at very low PAHs levels, and it was not till we got into the interior side of it and a little bit further that the PAHs levels started coming up. So we did this from several different sides, and we concluded that the PAHs were in the interior but not on the outside. And that was one of our lines of argument, that these PAHs were indigenous to the meteorite and did not come from Antarctica. Well, then we wanted to move on and look at this material some more with the scanning electron microscope, and we were intrigued with these surfaces that had the carbonate globules in them, and we started to study these surfaces. And again, the pancake-shaped form here, I'm going to show you a couple areas. One is in the center or the core of the globule, and then the other one is out in this dark band, which is the iron-rich band. Now, when we look at the center, we see, at this scale, we see a lot of nice crystal faces. These are clearly related to the crystallography of the carbonate, and we don't see much else in this region. We just see nice straight lines. But when we go to the exterior band at roughly the same magnification, we see that there's quite a difference in texture, totally different texture. We don't see any more of the straight lines, the cleavage forms of the carbonate. In fact, we see a jumble of other forms, some of which are rounded, some of which are elongated oval. Now, this is also the region where we saw the magnetite. So some of these guys are probably the magnetite. It is very difficult with the SEM to analyze at this scale at 100 nanometers or less and tell what you have. So we're not sure if any given particle is magnetite, but we suggested in our original paper that some of these particles might be, in fact, nanofossils or tiny fossils from Mars. And we see occasional longer ones here, here, and here. And those of you who were here yesterday saw some other pictures from another lab in Arkansas that independently found a lot of these guys on the carbonates where we're seeing them today. Now, again, we had to understand what these were. And our first innocent suggestion that they might be fossils of tiny bacteria or microbes from Mars raised a tremendous stir of controversy, and much of the controversy was directed at the size, and other bits of the controversy were directed at the shape and the composition. But the argument goes something like this: if a bacterium or a microbe is too small, there's simply not room in it for the cell walls and the DNA and the lipids and the proteins and the molecules that are needed to process food, to make food into energy. There's just no room for that below the original number quoted to us was about 500 nanometers. That has worked its way down now to maybe 200 nanometers. But the theoretical analysis is that there's no room in really tiny things for this factory. And so we set about trying to find some really tiny things in a terrestrial setting where we could more or less show that they're living bacteria. And there are other people that have done this. Bob Folk is speaking later in the week, and he will discuss even smaller things than we've seen on Mars, which he interprets as tiny bacteria. However, we went to the record. This is some peloids, or little carbonate globules found in oceans and bays. Shavitz of University of Houston etched some of these. And there are a lot of things here that are well under a micron in diameter, which he interprets, long before our Mars work, he interpreted these to be bacteria. Bacteria fossilized. We went to some travertine from some hot springs area. This is from Italy. We got the samples from Bob Folk. And we looked, and again, the scale bar here is about a micrometer. So these guys are roughly a third of a micrometer, or 300 nanometers in diameter. Now, this is smaller than the original limit that was given to us by the microbiologist, but now as they lower that limit a bit, it would include these as okay things. And we think we can see things like cell division and so forth. We've also looked at these in TEM. There's no doubt that these are microbes in our mind. There's another class of features that is not very well known called biofilms. Biofilms are essentially organic films secreted by bacteria to help anchor them to their environment, or to hold them together in colonies, or even to help change the local microenvironment, the pH or the EH, to something more to their liking. They also are fossilized sometimes, and they're found in particularly stromatolites. way back to almost to the beginning of the Cambrian. Well, let's look at a couple of these. This is some biofilm in the travertine samples. This has been etched with acid, which eats away some of the carbonate. Biofilms are not affected by dilute acetic or hydrochloric acid, so they remain behind, and that's what this webby stuff is here. Here's a sample from the Columbia River basalt taken almost 2 kilometers beneath the surface in an igneous rock. This sample has bacteria, which are about a micrometer, and then they have this kind of filmy, very thin filmy stuff connecting them. That is the biofilm. It's holding them together. It helps with their environment. That's why they secrete it. Well, how about the Mars meteorite? This is the Mars meteorite. It's been slightly etched in phosphoric acid, but some of us can see a very filmy area here, which is partially covering some of the grains, and is only, the scale of this is about 2 micrometers across here. These things are only a few nanometers thick. This is another area of the same chip, which has been, again, etched in phosphoric acid. As we zoom in on this area, we see a very complex network here. These things, we have stereo views. These things stick up in stereo, and it's a very complicated three-dimensional network. Well, what do we know that looks like this? This is a biofilm, a recent biofilm from a stromatolite, and it is about the same scale, very similar in scale. This is especially freeze-dried sample to preserve the original textures of the biofilm. This is mainly carbon and oxygen and hydrogen. It's a polysaccharide composition. We also saw in some of our samples, this is now unetched, we saw stringy things here, which we interpreted, again, as part of the biofilm structure. Things that string out, they cross from one mineral to another. We interpreted that these were made by the bacteria that we also saw in the sample. Well, when we go back to Allen Hills, we see kind of a stringy material that seems to bridge across a couple carbonate grains here. Now this has been etched, and so we don't quite know what the original stratigraphy was here, but this may have been embedded within the carbonate, and we've revealed it now by etching. But it is clearly a very thin film, and it remains after the etching process. Now, we've all seen these features here. This is what we call the worm. It's about half a micrometer in length, a little over half a micrometer. This is well within the allowable size of microbes, if this is a whole microbe. We were accused that was some kind of artifact, but Valley, our co-investigator, did it using a totally different technique, a platinum replica technique, carbon platinum replica technique, shadowed, and then using a transmission electron microscope on it. And he found essentially the same thing, the same size, the same curlicues in his sample. So since these techniques are more or less independent, we concluded that there's no chance of an artifact here, of sample preparation at least. Now, what about those forms in known terrestrial rocks? Well, we see some things like this. And again, this is pretty much the same size as what we saw in Allen Hills. Now, is this a microbe or is this something else? We're not sure in this picture, but we also see in Allen Hills a bunch of smaller guys like this, which appear to be again, elongated forms, unsupported, unattached structurally to the mineral. And we see similar things in the Columbia River basalt. These are all little fragments, which we think we start to understand now. These are clearly biogenic. And these are the same size as the wormy guy in the Mars meteorite. Now, what are these things? Well, they could be microbes, but they could be something else. And let's see what else they could be. This is a view of the Columbia River basalt here. These are some big microbes, about 1 micrometer in diameter. But look at this little guy here. What is that? Let's look at that more closely. There it is. When we look more closely at that, we see that what that is a single microbe with a tail on it. And that tail is probably a flagellum or an anchoring member which holds it to a substrate here. And during its lifetime, this microbe used that tail to anchor itself so it wouldn't get washed away in the groundwater flow. And notice the size of that. That's only 200 nanometers wide by about 300 in length. This is down now in the size range of what we're starting to see, many of the features we see in the Mars meteorite. And we even have some TEM photos of these guys. And they have often quite long tails. And here's a bunch of them. The little black arrows point to a number of these bacteria, and most of them have either whole long tails or cut off tails. And we've been able to get X-ray data on some of those tails. This is the mineral ferrihydrite. We think that the original organic tail has been fossilized now with ferrihydrite, which is a common mineral replacement during early fossilization, and that's what this guy is. In fact, in places there's this tangle that looks like a cat got a ball of string, and that is mainly ferrihydrite. This is TEM view. Now we again went to the literature and other publications. This is Berge's Manual of Systematic Bacteriology, and it shows a particular bacterium here. And that bacterium is held down by fibrillar material, which interconnects the cells and holds the cells to the base. This is similar to our biofilm that we saw earlier, except instead of being a film, it's a series of crisscross filaments. This is another view from a paper by Francis Westall. And these are some cells that are clearly anchored to each other and to a substrate with these filaments. And notice the filaments, although the bacteria are about 1 micron long, these filaments are only 50 nanometers or so in diameter. And occasionally, there are occasions where the filaments are partially buried by either biofilm or fossilization processes, and all we see are hints of them barely sticking out from a matrix. Sometimes in Columbia River basalt, we see that they branch and divide and make a fairly complex network. Now, when we go back to Allen Hills again, we have started to see now filaments in Allen Hills. This is in a part of one of the chips, and this is a filament that's over a micrometer in length. We think that it's been perhaps cut there and partly buried here, but that is a continuous filament in Allen Hills. And it is exactly the same size as those that I've been showing you. And in places, other places, we see hints of these filaments here. And then look at this. This is an area where we see essentially branching and filaments that are become partly buried here and become somewhat exposed here. This is a complex area, but we think that a lot of it is made up of these filaments, partly buried, partly fossilized is our interpretation. We believe that these filaments may be the same thing as what I've been showing you in terrestrial samples. Now we have another class of material in Allen Hills that we don't really understand. These are what we call the swimmers, and they always occur typically in patches where there's a whole lot of them, and we vacillate on what they are. They could be other forms of the biofilm filaments that I talked about earlier. On the other hand, these could be some kind of weathering product from the underlying pyroxene. As the pyroxene starts to turn into a clay mineral, these could be the edges of the pyroxene plane. We haven't made up our mind yet. We have not published these because we're uncertain as to their interpretation, but they may turn out to be important, and we're studying them right now. Well, let's look at some of the other features. We're moving along here. These are fossilized bacteria from the geologic record on Earth. This is from a publication by Bill Schopp and others, and one of the main features here are long, skinny filaments, filamentous bacteria. They look very similar to some of the filaments that we have seen in Allen Hills, except that these are much larger. They're 1 or 2 orders of magnitude larger than what we've described in Allen Hills, and that's created a bit of a controversy. How can these things in Allen Hills be fossil bacteria when the fossil bacteria are so large? Well, again, we're working now with several other investigators. This is Francis Westall in Rome. These are bacteria. The longest one is about 2 micrometers here from very old sediments found in the Ross Sea. The sediments are over 100,000 years old, and they're just full of these guys. The false color's been added, but they're clearly bacteria. That's about 2 microns, and this is 1 micron, and so forth. One thing that Francis Westall did was look at a very old rock, the Koenigsberg rock from South Africa, 3.5 billion years old. And she found a lot of these guys here. These are essentially silica, pure silica now, and again she's given them false color so you'll see them. These are on the order of 1 micrometer in length. And these are 3.5 billion years old. I think we're getting now into a region that has not been previously explored. That is really tiny fossils from the Earth's record, which are getting down below optical microscope range. Well, I want to finish up with some view graphs here, including some recent material. So if I could, I'm not sure how to. I have to.
Just tell me what graphs you wish.
Yeah, start here and just go through. Okay, we're having a little help here from Richard. This is, again, an area on the carbonate rim in Allen Hills. And I'm sorry there's no scale on it, but this biggest guy here is about 200 nanometers in length. We have seen a number of these clam-shaped forms. We don't know what they are. We think they're not minerals, but we're not quite sure what they are. They're always in a pair. These are 2 here. Next slide. Also notice some of the elongate oval things, which are about 100 nanometers. These are the same size as many of the things I showed you from the terrestrial record. And here are a couple more of these things. One possibility is that these are fossilized Martian bacteria which are in the process of dividing. One can almost see a seam or a crack here, and they may divide longitudinally, and so these may be the dividing pair. We're not sure of that, and there's no way that we can tell that. but it seems like a reasonable hypothesis. Next, please. Now, this is some fairly recent work. What we do with the scanning electron microscope is put a chip in there after lightly coating it with either carbon or gold palladium. Next slide, please. I'm going to zoom in on a central area here. And as we zoom in, the scale now is still quite low power, but there are some features here that I want to call your attention to. These features are these darker regions here. Notice there's some dark splotchy stuff in many places on this surface. It seems to follow the cracks to some degree. Next, please. Remember that many of these cracks are pre-existing. They were formed on Mars. As we get a little bit closer, we can see now the development of kind of a webby texture here and a very complex texture here, including some semicircular forms here, all associated with these cracks. And the scale here is, I think, 2 micrometers. All right, let's move in. Next slide, please. As we move in more on that area, we see several things. First, we see that there's kind of a big structure here that is on the order of a micrometer. And then we see that there's some of this filmy, webby stuff. And is that biofilm? We don't know yet, but we do know that it is carbon rich. We have that analysis. And then we see a whole lot of these little guys, which seem to be more or less clumped together here. And at this point, we don't really know what they are, but they're probably high mass because they have high electron emissivity. Next, please. And then we have, in the same general region, we have something like this, which doesn't look like much, but this is all carbonate, basically. We have the analysis that this is carbonate. It's a combination of magnesium and iron, calcium carbonate. And if you look closely, you can start to imagine little forms here that are about 1 micrometer in diameter. And next slide, we're going to look at this one a little bit more closely. And what we're suggesting is, and again, this is very tentative. We have to check this all out with careful mapping. But we're suggesting that this may be a fossilized microbe here. And this particular structure here may be the cell wall now turned into carbonate crystals. And that kind of fossilization is, in fact, relatively common in the terrestrial record, where these cells are replaced by carbonate, but you can still see a hint of the original shape and a hint of the cell walls. So we're going to pursue this. There's another feature about these that's quite interesting. Although they're mainly carbonate, they're relatively high in sulfur, and they contain some phosphorus. And we're in the process of mapping the sulfur and the phosphorus to see how it corresponds to the shapes of these guys. So I think the conclusion is that we're moving ahead, we're finding new data. We've had to go back and look at the geologic record on the Earth and start to build up a database of Earth features involving microbes, involving fossilization, involving biofilms, involving filaments and flagella and other kinds of appendages. We're doing that. At the same time, we're moving ahead with Allen Hills. We're finding new features rather than looking at the same old things. And we're really confident, more confident, that we're getting closer to the truth about these rocks. So I'll stop at that point. Thank you.
Thank you very much for that very, very interesting presentation. We have time for some questions. Do we have any questions? Yes, back here. Would you please, I think, are the aisle mics still on? Please come to the aisle mic or stand up and speak loudly.
Could you summarize how they changed the model so it would allow the ejection of material from Mars to survive intact?
They came up with two new ideas. One is that near the surface of the ground, the shock wave, the reflected shock wave coming back up, has nothing to push against when it's near the free surface. And so anything that is entrained in that shock wave, since nothing is pushing back, is able to be accelerated very rapidly without getting melted. That's one modification. A second modification is that they allow now for possibly more vapor generated by the impact, and that expanding vapor cloud can accelerate these rocks up to 5 kilometers per second. They even have a third idea, which is that an oblique impact, the momentum is transferred directly to some of the near-surface rocks, and these are thrown off into space. Again, this free surface effect prevents them from being squashed. And so there's not a consensus of models, but there are 3 new models that didn't exist before, each one of which can get these things off of Mars.
Can you be sure that these are not fossils from bacteria that was introduced to the rock once it was deposited on Earth?
That is a very important question, and we're attacking that along several lines. We're looking at known bacteria in Antarctica, which the dry valleys have quite a bit. We're trying to catalog that and its features. We're looking at a lot of other meteorites from Allen Hills and other locations, many of which have been in the ice much longer than Allen Hills. Allen Hills, by the way, there's data developing that has been in the ice. It's been on the surface only about 1,000 years. It's been in the ice for 10,000. So far, in looking at these control meteorites, we've not seen anything like any of this I talked about. And if you heard Paul Benoit's talk yesterday, he independently has looked at a number of meteorites and again found nothing like what he found in Allen Hills. It's very difficult to prove a negative here that there's no contamination, and we'll probably never totally prove that, but we're well aware of the problem and we're continuing to attack it.
We have another question
I'm just wondering how big the meteorite from Allen Hills is, 8401. Physically, how large is it?
How large is it?
Yes.
It's 2 kilograms at about the size of a baked potato.
Thank you.
Incidentally, we spent 2 years studying 3 grams out of those 2 kilograms. And we still have some left, but we are getting a new supply soon.
Is there any chance of seeing transverse cross-sections of your presumed nanobacteria?
We're still trying that. I showed you one cross-section of these little filaments, which are only about 20 nanometers in diameter. We were able to make that cross-section because we had a clump of them, so it didn't really matter where we sliced through. But it is an extremely difficult job because you first have to find them in the SEM and then transfer them to the microtome and slice them up and put them in the TEM. So although we're trying that, it is not clear that we'll succeed.
Okay, I think we have time for one more question.
Dr. McKay, is this the first time you've reported the objects you describe as looking like the cell wall and the splitting ovoid or whatever it is, and have you submitted these for publication yet?
No, we have not, and I'm hoping that information will not go outside this room so that we don't compromise our publication. Yeah, we do want to publish those, and we don't want to give them to the press right now.
Here I am.
Thank you very much. Well, thank you very much, Dr. McKay. The next speaker is Professor Thomas Gold. Thomas Gold is Professor Emeritus of Astronomy at Cornell University. He founded the Cornell Center for Radiophysics and Space Research, where he served as the director for 20 years. He received a Doctor of Science degree from Cambridge University. Dr. Gold has been a Robert Wheeler Wilson Professor at Harvard University and Chief Assistant to the British Astronomer Royal. He has served on the President's Space Science Panel for 7 years and received the Gold Medal of the Royal Astronomical Society and has been awarded an honorary MA from Harvard University. I present Professor Gold.
Thank you, Richard. I'm very pleased to have been invited here. The opinions that people have about what I have said vary. Okay. Now, following the previous speaker, I just want to say that I believe he has a very, very strong case on somewhat different grounds from those that he mentioned. I don't want to say that the grounds he mentioned are weak, but I think that the case altogether is still a lot stronger than comes from the previous discussion. The first thing that I want to stress is how common hydrocarbons are in the universe, in the solar system, in the molecular clouds in the galaxy, on other planetary bodies in our system, etc. Many people are not aware of that. I've been asked at conferences when I said, well, look, there are all these planetary bodies that have got hydrocarbons on them. People have said to me, but are there any other astronomers who believe that? Because there's a huge superiority in numbers of people who believe that hydrocarbons can only be made by biology. So I'll have the first slide, or I'll do that myself. Yes, can I have the lights a little lower? Can I do that? Can I have the lights? Yes, okay. Because so here we're going to look quickly at planets, and you see methane on Jupiter, in the atmosphere of Jupiter, but you will see it also in the atmosphere of Saturn, much the same. But also on Uranus, you see a comparison spectrum in the same waveband of methane and the Uranus spectrum, leaving no doubt whatever that methane is common. The same is true of Neptune. On Pluto, there's not much of an atmosphere, but the frozen material on the surface still mimics a mixture of methane and water ice, which is the curve below. And it mimics it pretty closely. On Triton, which is a satellite of Neptune, the same is true as on Pluto. It matches quite closely. So all these bodies have undoubtedly got at least methane in substantial proportion on their surfaces. The next one is the most interesting one of all, and that is Titan. Because it has an atmosphere, the ability to detect spectral lines is, of course, greatly improved. And you see the high peak of methane, but you also see other hydrocarbons, C2H2 and C2H6 and C2H4 and C3H8 and even other things like HCN. So on Titan, where we can really investigate the matter, there's a whole range, and indeed the clouds on Titan are chiefly made of methane and ethane. And presumably to keep these clouds in existence, there have to be oceans or large lakes, surfaces of liquid methane and ethane on the surface of Titan. Now, there used to be the conception, and many people still have that hydrocarbons cannot come from deep down on the Earth, for example, or on any of these bodies, because they will dissociate at a high temperature. And we have articles, learned articles written that say we cannot look for hydrocarbons on the Earth at deeper than about 10 kilometers. But in those articles, the word pressure does not appear. While, of course, the thermodynamic calculations completely change the situation once the pressure gets high enough so that its binding force due to the pressure is comparable with any binding forces of attraction within a molecule. Once you reach that limit, then, of course, you will have essentially a big molecule composed of hydrogen and carbon. And if you then let that, well, you will not need any special catalysts or any special reaction to set up hydrocarbons. Anytime you put hydrogen and carbon together at maybe 40 kilobars and 1,000 degrees C, that's we're talking 150 kilometers down, not 10. Anytime you do that, you don't have to bother to make them. They make themselves. This is just the thermodynamic enforcement of minimal volume. And what you see when it comes up is hydrocarbons at every level. As you go down to lower pressures and lower temperatures, there will be stability points, virtual stability points for different components. And it is those, a mix of those that accumulated on the way up, that you then find in the shallow wells that we drill. This diagram is meant to indicate that. I can't see from here, but you can see it. I can't see whether you can see it.
Ah, yes.
It's a little dim, but yes. Okay. This is depth in kilometers, one side or other. I can't from here read it. And this is temperature. And you see this is the usual PT domain from a depth of 300 kilometers up to the surface. And these lines here indicate which types of hydrocarbons would form from a random mix of hydrogen and carbon. And many of them, or perhaps most of them, would then drop out and become stable out of a general stream of methane that carries them up. Methane is the most abundant of any of the hydrocarbons. Carbon is the fourth most abundant element in the universe. Hydrogen is the most abundant. It's a very near-fetched idea to think that hydrocarbons are the most common, but not just for that reason, but because we measure hydrocarbons in the molecular clouds, in the comets, on all these planetary bodies that I've shown you, in the dust grains which Richard Zare so cunningly analyzes for the polycyclic aromatics. It is just absurd to think that just our Earth did not get any hydrocarbons built into it when it formed, when all these other bodies evidently did. Now, carbonates are not common in meteoritic material. So if one discusses what the materials were that made up the Earth or Mars or whatever other planetary body, carbonate is not a very likely guess, because we're pretty sure the meteorites are some kind of random set of leftover debris from the formation of the planets, and evidently in them hydrocarbons are much the more common form of carbon than carbonates. Now, then I want to tell you that the carbon budget that we can make of the Earth means that one needed to supply, in the course of geologic time, roughly 2,000 times as much carbon in order to build up all the layers of limestone that the Earth possesses, 2,000 times as much as is contained in the ocean atmosphere system at the present time. Now, it's unthinkable we're just arriving here on the Earth on the last day, so to speak, that it can lay down limestone. So the other explanation is, of course, that it has been constantly resupplied. If it was resupplied, my guess would be it would be resupplied as hydrocarbons, because the atmosphere will very, very quickly turn it all into CO2, and the CO2 will readily combine with metal oxides in the ground to make carbonates. There are more detailed arguments in existence of all this in terms of the quantities of oxygen that exist, and they all point in this direction, that you must make at least a major fraction of the stuff come up as hydrocarbons. But you see, in geology, the idea that gases or liquids or fluids have come up from below is admitted only insofar as lavas are concerned. Beyond lavas, they allow you to have maybe dissolved gases in the lava. But here you have an explosion like Krakatau or Mount St. Helens or something, enormously fierce explosion. And then the explanation for this is to be that this exsolved from the lava when the lava came to lower pressure. That would be a terribly slow process. No, there is plenty of gas in the Earth, and the diamonds clearly demonstrate that, because the diamonds are must be at a depth of 150 kilometers before the pressure is high enough. There must therefore be fluids down there that concentrate highly concentrate clean carbon, which then crystallizes out into diamonds. And then there must be the huge explosions that have brought diamonds up from great depths to the top. So we know that there's unoxidized carbon in large amounts down there, because we do need essentially methane or possibly ethane to lay down the diamonds. Now, I just want to read you out the sentence from Robert Robinson, who was a very famous chemist. Arguments based on composition fit equally well or better with a primordial hydrocarbon mixture to which bioproducts have been added. And that is the message that I will try and put across to you. Mendeleev had already said the same thing 100 years earlier, for much the same reason. Now, where do hydrocarbons occur on the Earth? I need all this in order to explain to you why I think it likely there is life on the Martian meteor, was life on the Martian meteorites. Where do hydrocarbons occur on the Earth? Well, the best example that the hydrocarbon occurrences span over very much larger regions than the surface or near surface topography or geology. The best example is the island arc of Indonesia. It's delineated sharply by the numerous earthquakes that happen. There are many places that have several earthquakes you can feel per day. There are 8 earthquakes of magnitude 8 in this century. So it's, California is nowhere. And you see that it's marked in gray is the earthquake belt, and it goes through the islands of Indonesia, Java and Sumatra, etc., and then goes through the Andaman Islands in the Indian Ocean, and then up into Burma along the Irrawaddy River. Now, the reason I'm explaining all this to you is that so do even the monstrous earthquakes, but also the active volcanoes at the present time, which again go right up into the mountains of Burma, and so do the hydrocarbons. And that's, I say, is a point that Mendeleev had stressed, that obviously the scale of this is much, much larger than any geological scale. So that also points to them coming up from deep. Now, then there is another item, which is the vertical stacking that has been much discussed. The vertical stacking of hydrocarbons concerns the situation that if you drill in one place and you find a hydrocarbon, go on drilling, there'll be more below. Go on drilling even if it means drilling into bedrock. It is now thought that more hydrocarbons have been commercially obtained from bedrock, from crystalline rock, from the porosity that they sometimes possess, more per foot drilled than per foot drilled in sediments. So the idea that you need sediments to make oil is out the window. So we have the hydrocarbons on the Earth for just the same reason as the other planetary bodies, and why shouldn't we? Now then, I want to show you a helium, nitrogen. No, first I want to do this one. This is the vertical stacking in South America, and this is an oil map of the Sao Paulo Basin in South America, in Brazil. And look at the green patch, if you see what I mean. This S-shaped or double S figure there, and memorize it for the next picture. This is coal, bituminous coal at one level. No, I'm sorry, this is oil at a deeper level, and this is bituminous coal at a shallower level. Completely different age when it was laid down, no connection that one could trace through the geology, and yet it's clearly the same region. Well, there are hundreds of places on the Earth that I could show you that have quite the same situation. There's hydrocarbon in Oklahoma as oil and gas at deep levels, as coal at shallow levels. The same is true in Iran in very large quantity, even in Saudi Arabia. Now, I believe that bituminous coal is not solidified, is not carbonized wood or any of these things. Very few fossils in bituminous coal, much less than in the surrounds. So that was just an invention, I think. It is essentially a hydrocarbon, of course. It's largely soluble in oils. You can take a lump of bituminous coal and mix it up in oil. If you mix it up for long enough, you'll get about 3/4 to become liquid in the oil. So this is vertical stacking. This is another example of vertical stacking in the Four Corners region, in the San Juan Basin. And you see from deep levels where you have gas and oil, gas there. And then you see, then you come to coal up here, And there are many such sections that have been drawn, and they all show that. Well, the only way that you can imagine getting a vertical distribution of hydrocarbons is by them coming up from below, because they're not going to go down from above. You cannot say that it was because biology was in some way particularly favorable, because much of this is below any biology or any other biological traces. So, all right, if we say all that, then we come to another problem. And the other problem is that we have big areas on the Earth where we have ratios of hydrocarbons, nitrogen, and helium closely associated. Most helium on the Earth is in connection with hydrocarbons. All the commercial production of helium is from hydrocarbon wells. Within the hydrocarbon well regime, the helium is most abundant in mixed oil-gas wells than it is in pure methane wells. But it is also, in pure methane wells, much more abundant than in the surrounding rocks. How can this come about? Well, what you see here is a large region that has a common ratio, it's a straight line, a common ratio of helium to nitrogen, is plotted this way. But if you plot this way, it would be methane that you would be plotting, because there's not much else in this gas other than methane and nitrogen, and the helium, of course, is only a few percent. So you have a relationship between helium and hydrocarbons. Well, that is itself is, of course, indicative that the hydrocarbons have come up from deep down below, because it's only then that you could understand that it would preferentially sweep up the radiogenic helium that is made in the rocks, while if it had come from above, there's absolutely no way in which you'd concentrate the helium. The helium you can only concentrate essentially with a mechanical pump. There's no other way. It is not chemically concentrated, doesn't form any compounds that would chemically be concentrated, or anything of that kind. So a mechanical pump, the only reasonable exposition of that I can think of is for fluids to have come up from great depth and washed through all the channels through which they have traveled and picked up whatever helium was made available in those channels by the radioactive decay of the uranium and thorium in the rocks. All right, then we can understand all this then if we make the hydrocarbons so easily at great depth and then have them seep up through the rocks, as of course they would, because they're buoyant relative to the rocks. Then we'll have the helium concentration. And I can tell you that there is no English-speaking literature that discusses this possibility, and none that discusses why helium would be associated with hydrocarbons. In Russia, there's an awful lot, several hundred articles that I have in my reprint file, for which I paid for the translation. In this country, it is just considered impossible that any hydrocarbons have come up from deep down, and that's the end of it, and the helium can't be explained. And I get remarks such as, oh, well, I'm not an expert in helium, or the helium is in such small quantities, we can't tell how it got in there. It's not of importance whether it's in small or large quantities. It is important that there's more in the petroleum than there is any place else. So it has clearly been concentrated. But then the moment that you say that, then you seem to be in trouble about the biological molecules that undoubtedly are in petroleum. And they used to be explained by saying that, well, this stuff has come from biological material. But if it comes up from a depth of 150 kilometers, it's not going to be coming up from biological material, from 1,000 degrees C. So then, it was at that point that I realized that we must expect a huge amount of subsurface microbiology. Only in that way could we understand that the hydrocarbons have washed up the helium and thereby greatly concentrated it, compared with places that did not have any washing up of hydrocarbons. And then at the much shallower levels at which we find oil, down to perhaps 10 kilometers, at those levels the temperatures are not too high for microbial life. You're talking now of 100 degrees, 120 degrees C or something of that sort, maybe as much as 150. And those are temperatures in which it is largely known that you can have microbial life. I was responsible for drilling. How much longer have I got? Huh?
30 minutes.
I was responsible for drilling 2 deep wells to depths of 6.5 kilometers or so in Sweden, in the granitic bedrock of Sweden, but in a region in which it was shattered because it had been very long ago a huge meteorite impact. Of course, it's completely cemented itself together again, and it's essentially a solid, but perhaps with slightly more cracks running through it than would have been there without the impact. And this particular region had a huge abundance of hydrocarbons on the surface. You could get oil dripping out of a rock and fill a beer bottle with it in half an hour. You could get it out of very shallow holes. You could find methane between 200 and 1,000 times more concentrated in the little lakes and ponds that were inside this crater than in similar lakes and ponds and similar woodlands on the outside of it. So it was very clear that indeed stuff was streaming through even now. And this now is 2 billion years later than when the crater was formed. So there was very little doubt that this was an interesting hydrocarbon area, but of course the conventional theory said that there could not be a stitch of anything of that kind down there. You should know that the oil in the Middle East was discovered by people trudging through the desert and picking up their boots dripping with oil. It was not discovered because there was any geological evidence for the oil. On the contrary, the people who investigated the Middle East advised the then oil companies not to purchase any of the franchises for drilling there because there was not a significant amount of biological material buried in the sediments. And these are the world's largest deposits now. So I don't know, I find it terribly strange that they will want to stick to this story after what we now know. Now, so when I realized that one had to explain the helium concentration, and one had to explain at the same time the concentration of clearly biological molecules, but like Sir Robert Robinson had said, they look much more like a contaminant that had been added to non-biologically made oils. And he had said that because biologically made oils will be short of hydrogen. The hydrogen-carbon ratio, if you let some stuff decay, will be about 1 to 1. You cannot get anything like 1 to 4 out of it, or even 1 to 2. So he and also Mendeleev already 100 years earlier had recognized that. But on the other hand, since there are clearly biological molecules in it, Robert Robinson had said the right thing, it seems to me, that there must be a location where hydrocarbons are added, biomolecules are added. So that brought me then to writing the paper, The Deep Hot Biosphere, in which I proposed that the hydrocarbons were coming up from deep, and there was a huge amount, and you need a huge amount of microbiology sitting underground, so that every oil well of the thousands that exist on the earth has been contaminated. And those are not only the very productive oil wells, that includes thousands of others that for some reason cannot be made to flow freely, but nevertheless contain an awful lot of oil. Oil is enormously abundant on the earth. And if you include if you include methane, then it is by far the most abundant form of carbon other than the carbonate rocks. Even just the ocean deposits of methane hydrates, of methane water ices, amount to more than all coal and all oil and gas put together. And that's on the ocean floor, where you have virtually no sediments underneath, lying more or less directly on top of the basaltic ocean floor. So hydrocarbons on the earth are very, very abundant. Now here, if you can see this graph, is a depth range in one of my wells in Sweden. And this is in meters. And you see the white line is the abundance of methane that we observe. The yellow line is the abundance of free hydrogen that we observe. This red line is the abundance of methane, and the others are of two other hydrocarbon gases. Now, what is interesting about that? Some people have said, oh, well, this is all meaningless because it could have been a contamination from above, because you've used drilling fluids that contain diesel oil and things like that. In other words, they said you poured it in from the top, because it's not possible for any oil to be down there since there's no biology down there. That was actually in a referee's remark who accused us, a large company doing this, accused us all of spiking the well. I mean, and then I show them this, which was an observation done outside the company by by a company that does commercially analysis of hydrocarbon wells. And of course you see that there's no question of a correlation of the peaks and valleys of helium with the correlation of hydrogen, and indeed with the correlation of all the other hydrocarbon gases. Well, nobody has yet succeeded in spiking a well with helium. Now then, the most conclusive item that I have is a depth range in one of the wells, and it shows you the large peaks of hydrocarbon content in igneous intrusions and in the immediate vicinity of igneous intrusions in the granite. The igneous intrusions, of course, were much too hot to carry hydrocarbons up with them to these shallow levels where the pressures are rather insignificant, like 6 kilometers. But apparently the pathways that were either created by the igneous intrusions, and they certainly came from below, or that were used but not completely filled by the igneous intrusions, these pathways are apparently active to the present day in supplying hydrocarbons just there. See, what I've marked here is the regions in which we find in the drilling chips igneous rock. And you see this one corresponds to that, this to this, this to this. And I've drawn two red lines where there was very much of the igneous rock in the chips and one where there was little, and you see the correspondence just could not be more precise. In other words, this leaves no doubt that the hydrocarbons that we see in that whole area has come in the same pathways as the igneous rocks, and therefore it has come from deep below. Now, so for this reason, I stressed that there must be this large amount of microbial life below. Now, the general viewpoint had been that we drill a little hole from the top, just a water well or something, and we look at the density of bacteriology, and it drops off sharply from 2 meters below the surface. If you go down to 20 meters below the surface, you're down to less than 1% of the density of microbes. And so this was extrapolated to say, well, much below that, there can't be any. Now we see just the opposite. We see that the substances that have come up from below serve indeed as the food for an extremely rich microbial life. And that, of course, is then concentrated at the levels at which it first can grab hold of its food, where there's the best supply, because every microbe is going to work its way towards the source of the food, or their progenitors will. And so it's at the deep level where the temperature gets to be perhaps somewhere to the limit where biology can work, that you find the most. And you find enormous quantities there. So in my Swedish wells, I invited the Swedish National Laboratory of Microbiology to go and sample that well with many samplers at different levels that could open and shut, and to analyze and see whether they could culture any microbes. And the answer was that they could culture microbes. Is Mr. Sheftsik here? No. He was the prime mover in this, and they could culture them. They were not the primary microbes, as I believe would be there, but they were microbes that were concerned with fermenting other microbes that lived on the primary materials that were available. Now, what were the materials that were available? Hydrocarbons that get delivered on their doorstep, like milk, that stream through. You see, it is of interest that for microbiology to thrive in a surrounding like that, it has to have a constant supply, or reasonably constant supply, of food. If you merely had a mix, a limited quantity of a mix of liquids, fluids, they would exhaust that and then it'd be finished. But if you have a situation where a fluid streams through the cracks, and where the cracks of the rock provide another component with which the oil can be oxidized, then of course they have a very good food supply. Now the components that they would have to take from the rocks in order to oxidize the hydrocarbons are loosely oxidized, loosely bound oxygen in oxides. And what is there? You can calculate that most of the metal oxides that are in the rocks have the oxygen too tightly bound to make a go of the transaction of taking it off from the rocks and using it to oxidize hydrocarbons. There are really only two types of substances that are really abundant in the rocks that can provide loosely bound oxygen, and that is highly oxidized iron, like Fe2O3, rust, which can be turned to Fe3O4, magnetite, and the other is sulfates, oxidized sulfur compounds, which can be reduced and the oxygen taken off into sulfides. And these being the two major ones, I would then judge, and that's what I proposed, that you would find magnetite and metal sulfides as a major component of the leftover stuff, which of course would also contain leftover oils. Because in this case it is the oxygen supply that is likely to be more limited, not the hydrocarbon supply. And then there's a fourth substance that you would like to find, well, you would then find in association the oils, the leftover oils, the sulfides made from the sulfates, and the magnetite made from Fe2O3 or higher. But there's a fourth substance that you would also expect to find, and that we do find in oil wells on the Earth. And this comes closer to what my colleague in the previous lecture has talked about. You would find that you would have methane be oxidized and turned into CO2 from any oxygen that can take off the rocks. And CO2 in rocks will, in many cases, quite readily convert into carbonates. And when it does so, it will do so with an isotopic signature, which is very clear and very evident, and I hope that this will also be examined for the carbonate rocks in the Martian meteorites. And the reason is that the methane is the only carbon gas that can be fractionated very readily, because it has a 3% difference in diffusion speed. So if you have a diffusion barrier somewhere, you can get a large fractionation happening. The fantastic idea has become established that only biology can fractionate methane or carbon in any way. Of course, that's nonsense. Biology only does it with a semi-permeable membrane, and there are plenty of diffusion membranes that are nothing to do with biology. Even just diffusing into water and out of water is already a very large fractionation step for methane, but not for any of the heavier compounds. The CO2 you can't fractionate anymore because the difference between whatever it is, 46 and 47, is too small to make any difference to this. But the difference between 16 and 17, which is methane with a heavy carbon isotope, is big enough to make a large fractionation. So then you find in the carbonates that have been water deposited and that are crack-filling carbonates, just as Mr. McKay discussed, those show a huge spread in the carbon isotope values, while other carbonates do not. So having these four substances closely together seems to me to make a very strong case that this is indeed the right explanation. Now, I want to stress again that I think many people have thought that the presence of the oil of the PAHs was itself a strong piece of evidence for biological production of hydrocarbons. I think that is quite erroneous. The hydrocarbons are present for the same reason on Mars as they are on the Earth and as they are on Titan and Triton and Pluto, etc. And they keep coming up from below and they keep supplying our atmosphere eventually with CO2, where their pathways on the Earth takes them through volcanoes. They get oxidized in the lava into CO2. Not all of them. There's sometimes as much as 17% methane coming out of active volcanoes. Where a huge volcanic eruption occurs, there isn't time and there's not that much surface area for a gas bubble that is shooting up through a lava channel to get all oxidized. And the big eruptions where there's nobody there to measure the gas, or if there was, he ain't talking. As there was at Mount St. Helens, you know. They don't see what happens at the big eruptions, of course, but we do know something about big eruptions. There are many big eruptions where there were gigantic flames standing on top. I'm not discussing the spray of hot lava, as often has been said, oh, well, nobody can tell the difference between the spray of hot lava and flames. No one can tell that difference perfectly well. If there's a suitable wind, then you can get the flame completely clearly exposed and you see a blue flame. But even better than that, Krakatoa, between Java and Malaysia, at the end of the last century, had many eruptions that were completely confined to below the ocean surface. And a huge area of ocean surface on top just had dancing flames on it. So it was bubbling, it stopped all the volcanic ash, of course, coming through the water, but it did not stop the combustible gases coming through. So the reason why I say all this is that I expect that on many planetary bodies, the subsurface conditions will be quite similar to those on the Earth. I believe that the impacts that liberated what must have been tens of thousands of meteorites from Mars, for the 6 or 8 to be discovered in Antarctica, of course, would have been quite large an impact. And in a large impact, what happens is you dig a very deep hole and you throw out a lot of stuff that has not been heated very much along the edge of that surface, of the final surface that the crater will have. Most of that material has come from great depth. It does not matter what today's or what yesterday's surface conditions were on Mars. I do not myself believe that there could have been liquid water on the surface of Mars. It would either be frozen or evaporate. But it is of no concern because there will be water down to some depth it will be frozen, and beneath that it will be liquid, because any body the size of Mars will undoubtedly have a positive temperature gradient with depth, and so you must come to liquid water. Probably you will come to liquid water at a depth as shallow as 1 kilometer. And so probably many features that you see that look like stream beds exist also on the moon, very similar looking. And I think that those are just the infallen top kilometer when an underground river has made a curlicue channel. Water will in any case be present on a body like Mars, because it is just so abundant on the planetary bodies altogether that it would be very strange if Mars did not have a lot of water. But in addition, the oxidation of hydrocarbons on the way up will of course make water. So you will find CO2, which is largely turned into carbonates, and you will find water, which may well be in sufficient quantity to sustain the life of other bacteria. So you have all the circumstances underground, almost certainly just as good on Mars and on many other planetary bodies as you have on the Earth. So since by now people say, oh, it was just a conjecture when Gould first wrote about life deep on the Earth, it was not just a conjecture. It was the only way in which I could see that helium and biological molecules could be found together, because neither will attract the other. And then I had to have the subsurface biology. Well, we now have seen plenty of examples. In the last few years, people have examined many oil wells and so on and found a huge quantity of microbial life. One has not yet done everything about it, namely any microbes that live on the gaseous hydrocarbons. One cannot reproduce, one cannot culture in the lab, because one would need a huge pressure. So one talks about pressures of 10,000 atmospheres or something like that. And nobody has a culturing equipment that can go to these pressures. And if you reduce the pressure to 1 atmosphere, well, then you've got a concentration different from what it was down there by some enormous factor, not quite 10,000, but by an enormous factor. And the bugs that are used to live on a rich soup of methane are not going to be satisfied if 1 methane molecule hits them every half hour. So that will have to be done. And then we should be able really to go to town and see what microbes and what they look like, and whether they're nanomicrobes or what. That will have to await really careful investigation with the circumstances as they are down there, and one will then see whether indeed these products that I mentioned, these 4 products, are present, can be expected, as I have said. So I'll close just with saying that the case for the Martian meteorites, which incidentally I proposed that the Martian meteorites would be interesting ones to look at in 1990 in a paper that I submitted to Nature, which Nature flatly rejected, and which I then published 2 years later in the Proceedings of the National Academy of Sciences. Now, I've brought some copies of this paper, and they're lying on the table here. It's called the Deep Hot Biosphere. There are also some copies of Life on Other Planets, which is quite similar to the book that you already will have. So that's where we stand on all this, and I believe that we have a very exciting time in front of us. And my advice to NASA has been, and has been for a long time, that this will become a central object in their planetary work, the microbial life, and moreover, that places that were at least originally deep should be sought, because you might there find material that was once in the liquid water domain, even if it is not now so. And you might then be able to analyze that for the remains of bacterial life. Now, if you do that, suppose we do that in one location, say on Mars. I mean, I would go to the deep valley there where you undoubtedly have landslides that have come from the liquid water domain. Suppose we find, maybe I should show you the next slide. These are microbes from Sweden. The chirality issue. Suppose we find on another planetary body the same basic chemistry as our chemistry. You know, amino acids and DNA and all that stuff. Then we would wonder, is it because it was transferred from the Earth, as of course as we now see it could have been, or to the Earth from Mars or whatever, panspermia as it's called, or is it an independent origin, for which case a similar chemistry is very likely. I can't say that it must be a similar chemistry, but you know, if it was a chance of 1 in 3 that it would be a similar chemistry, that there are only 2 other competing chemistries to make anything that propagates like life, then you wouldn't know the answer between those 2. But suppose you look at the chirality and you find that it is the same as the chirality of living matter. Now, you know, if you take a liquid, there are no liquids known on Earth that are racemic except biological ones. Somebody wrote a paper in Science recently, or Nature was it, to say the Murchison meteorite shows non-racemic mixtures, and this, he said, is the first clear proof that biology is not needed to make non-racemic liquids. How come that on the millions of liquids that have been tested on the Earth, not one has turned out to possess this when it was not biological? No, I mean, it seems to me make a very strong case for the Murchison indeed having contained in its parent body some biological stuff. So let me go through that last step again. If we find the chemistry the same, then we look at the chirality. If that is the same, then you say it's possible that it was panspermia, but of course we can't be sure. There's a 50-50 chance that the chirality would have hit off on the same. But in that case, at least we know that our type of chemistry is common. Suppose it was the opposite, the same chemistry, but the opposite chirality. Then you would know immediately an awful lot, because you would say, well, our chemistry must be a very prevalent one for any kind of living mechanism. But then it has a 50-50 chance of hitting off one or other chirality, and so it is not panspermia, and so it develops independently in these bodies. Wonderful. But in the first case, if it was the same, then as I say, there is still only a 50-50 chance of panspermia being responsible. Then we'd have to go to another planetary body. And if we a third time found the same chemistry and the same chirality, and maybe a fourth time, then we would feel pretty sure that it is panspermia. So far as the origin of life is concerned, this ladder of evolution of our science is going to become the most fascinating and far-reaching exercise, and we will, I think, one day, with the help of this, be able to understand in some detail that huge question of the origin of life. Thank you.
Well, thank you very much for that absolutely delightful and most interesting presentation. We'll take some time for a few questions. If anyone has a question, please go to the mic.
What do you think the likelihood that there would be evidence for biogenic activity found on rocks on the surface of Mars?
Insofar as Mars has a huge number of craters on it, there will be still a lot of debris that, like the Martian meteorite, but did not get blown off Mars, and it will have the subsurface life in it, you see. But the notion that there was liquid water and that the sun shone brightly and that the trees grew and all that, I do not take very kindly to that. You need a huge atmosphere before you have liquid water. You see, on Mars you need 3 times as much mass above each square centimeter before you will have a reasonable range of temperature between ice and vapor, between ice and boiling, you see. We have 100 degrees C under 1 atmosphere here. To get 1 atmosphere on Mars, you need 3 times as much gas per column. I don't think that Mars ever contained any such huge amounts of atmosphere, quite apart from the question whether the greenhouse effect could be large enough to get at 1.5 times the distance, enough energy to gather into the atmosphere to get to the freezing point, to get above the freezing point of water.
Yes, are there other questions?
Over here.
Okay.
Curious as to your argument about coal, that some of it, I believe you would say, was of biological origin,
Yes.
and much of it was not. Do you have any thought of...
Yeah, well, I can tell you exactly where the division comes. Lignite is, to a considerable extent, of biological origin. A large proportion of it is fossils. It contains a large amount of granular rock in it. You know, the proportion of ash is something like